Thank you very much, and thank you for the invitation to come and speak today. In addition to all those things, I'm also a mother of 3 school-age children, but they're not in high school yet. I have a kindergartner and 2 third graders, but they're in 3rd grade. They're already starting science projects, which is very exciting. Actually, I had to donate dry ice a couple weeks ago for a Halloween science experiment. So I'm going to talk to you a little bit about diabetes, and I'm going to talk to you about a specific research question that we're trying to address. And I'm going to try to make it relevant to people with diabetes, because it is very relevant. And then I'm going to, so it's going to be kind of a mixture. So if you get lost in some of the science and some of the cartoons and things, hopefully it'll all come back and it will make sense. And certainly happy to answer any questions that you have as I go along. Okay, so I think everyone knows that type 2 diabetes is reaching epidemic levels, and I really appreciate this figure because basically it's showing that over thousands and thousands of years we started to become more obese, and while there seems to be lots and lots of focus on the fact that we just eat way too much, I would argue that probably the major part of the problem, or a very large part of the problem, is the fact that we're very inactive. So a major factor leading to increased rates of type 2 diabetes is inactivity, and physical exercise has been shown now in many epidemiological studies to be critical in the prevention of type 2 diabetes. So these are what I'm going to try to cover today. First, I'm going to start off and give a little bit of background on diabetes. Then I'm going to talk about skeletal muscle glucose transport. And then I'll talk about some studies that we've done in human subjects. And then I'll talk about this idea of an exercise pill. Okay, so some basic physiology. Even when I talk to all sorts of scientists, I always start off with a slide, make sure we're all on the same page in terms of what's happening in the body. So basic physiology: glucose concentrations in the blood rise after a meal. Insulin is released from the pancreas, binding to insulin receptors on tissues throughout the body, resulting in glucose entering the tissues. Now, in type 1 diabetes, or what used to be called juvenile onset or insulin-dependent diabetes, the pancreas loses its ability to produce and secrete insulin, and this is almost always caused by an autoimmune attack and destruction of the beta cells of the pancreas. Now, type 2 diabetes is the other major classification of diabetes. It used to be called adult onset or non-insulin dependent diabetes. We don't call it adult onset anymore because we're finding that kids age 11, 12 are coming down with type 2 diabetes. So here, insulin is produced by the pancreas, and levels in the blood are often normal or even high, but the tissues don't respond. We say that they are insulin resistant. So why is this such a crisis? Well, it's because diabetes leads to numerous complications. In fact, diabetes is the leading cause of blindness in our country. It's a leading cause of kidney failure, of amputations, a major cause of heart attacks, and a major cause of strokes. And it's very, very much a problem, and it will continue to be a problem because it's really reaching epidemic levels. So here's a map of the world, and this is some WHO data. So these are data in millions. So in the year 2000 in North America, there were an estimated 14.2 million cases of diabetes. And by the year 2010, next year, it's estimated to be 7.5 million cases, or a 23% increase. And these dramatic increases in diabetes are not just in North America, but they're throughout the entire world. And the WHO estimates that by the year 2010, next year, there'll be 221 million cases of diabetes, and by that 2020, this will increase to 300 million cases of diabetes. So it's a huge, huge burden. It will be a huge burden on the healthcare of the entire world. Now, the news isn't all bad, and there have been some studies done. This one is called the DPP, Diabetes Prevention Program, that was done, and this work was published in the New England Journal of Medicine a few years back. So basically what this is showing is that they took people that were at very high risk for developing type 2 diabetes, and they either treated them with lifestyle by metformin, which I'll talk more about later, which is a major type 2 diabetes drug, or a placebo. And you can see that the people with lifestyle had a 58%, who had a lifestyle intervention, had a 58% reduction in the incidence of diabetes. And this lifestyle had a very large component of exercise. Okay, so this then, as a little bit of background on diabetes, leads me to talk about our major research focus in our laboratory, which is skeletal muscle glucose transport. Okay, so why can exercise prevent diabetes? Well, I think this is shown very simply on this graph, which I'll develop here in a second. So basically, if you take a person with type 2 diabetes and you have them exercise at 70% of VO2 max, which is considered a moderate intensity of exercise, this is what happens. Blood glucose concentrations can fall. So here, this is actually a mild increase in blood sugar or blood glucose concentrations, and that with exercise, the blood sugar levels come down and remain lower after exercise. So why does this happen? Why does exercise lower blood glucose concentrations? And the major reason is because the blood sugar goes into the exercising or into the contracting muscles. And this can happen in people with type 2 diabetes at either normal or near normal levels. Now, if we look, of course, insulin does the same thing. You eat a meal, insulin levels go up, and insulin then causes the blood sugar to go, the blood glucose to go into the muscle. However, as I mentioned earlier, in people with type 2 diabetes, the muscle is insulin resistant. The insulin doesn't work. So we have a situation then when we're looking at this very important glucose uptake into the muscle in people with type 2 diabetes. The muscle is resistant to the action of insulin, but exercise works normally or near normally to promote glucose uptake. And so a major focus of the research in my laboratory is to understand the molecular basis for the ability of exercise to increase glucose uptake in skeletal muscle of patients with type 2 diabetes. And this is our big research focus in the lab. Okay, so to do this, we use a number of different models, from cells to humans, animal models, and I thought I'd just mention some of these here. So one of the systems that we use is a cell culture system, where we can contract the muscles in cell culture. This isn't the best system to use because characteristics of these cells are not really like adult rodent or human, but we can use this type of system. We also can use a system to study muscle contraction, where we take small, thin animal muscles from animals, and we can do direct electrical stimulation of the muscles. We can then measure things like glucose transport or proteins in the muscle. We can also do experiments where we anesthetize animals, and we anesthetize them and we attach electrodes to their hind limb muscles, and we can do contraction by nerve stimulation. And then we also do a lot of exercise by treadmill running. So we have a rodent treadmill. This is a picture of our treadmill in the lab, where we can actually exercise rats or mice 10 at a time. And these can be used for training studies, chronic exercise, or they can be used for acute studies. We also can do studies in the lab of chronic exercise, where we can put animals in wheel cages and they will just voluntarily exercise. This is a much better way to exercise animals than using treadmill running. And believe it or not, these animals can exercise, or they typically will, let's see, take a guess how much a typical, say, mouse with legs this long will exercise in one day. Anybody have any thoughts, or how long it is, how much they do?
5 hours.
What's that?
5 hours.
5 hours, yeah, not in time, but in distance.
3 miles.
3 miles. 6 miles a day is how much they do, yeah. Okay, the legs this long. So you put them in there. Now it starts to, as they get older, it starts to trail off, but they'll still do about 2 or 3 miles a day. It's really quite fascinating how much, and this is both mice and rats will do about 6 miles a day. And then finally, we do studies in humans, and I'll talk about some of that work too, and how we do that. Okay, so how does glucose get taken up into muscle? This is sort of the hardcore science that we're studying in the lab. Well, it does so by this system called the glucose transport system. And glucose transport is the rate-limiting step in glucose utilization. So the glucose comes into the cell and it's metabolized, but really it's the transport process itself that's rate-limiting. So it's really key in this process. It occurs by a process of facilitated diffusion. So that means it makes use of carrier proteins in the cell membrane. These are called glucose transporter proteins. And there is a whole series of these transporters expressed in mammalian cell types. And the two transporters, GLUT1 and GLUT4, are the ones that are expressed in muscle. And GLUT4 is the major one expressed in muscle. And so you'll hear me talking a lot about GLUT4, this glucose transporter protein. And insulin, exercise and insulin are the most physiological stimuli that can increase glucose transport into the skeletal muscle. Okay, so how does this happen? Well, actually, what these glucose transporters, this GLUT4, sits inside the cell. So in a resting state, if you're just sitting or you haven't eaten a meal, you don't have insulin stimulation, the transporters sit inside the cell in these vesicles. These are highly specialized vesicles. Then with an exercise stimulus, you actually get a movement of these transporters to the cell surface where they fuse and then allow for glucose to be transported into the cell. And so this is what we call GLUT4 translocation. Now insulin does the same thing, except insulin works through its own specific receptor. So insulin, there's an insulin receptor that sits on the cell surface. Insulin binds to the receptor, activating some signaling processes that both cause GLUT4 translocation. So how do we know this occurs? Well, there's a couple different ways that this has been shown. One is by doing fractionation of cells, isolating intercellular membranes and surface membranes. But one of the senior postdoctoral fellows in my laboratory has actually recently developed an imaging system to actually view this in muscle. And here's an example of this. So what he does is he takes an animal, it's a live animal, and he transfects it with this GLUT4 glucose transporter, transfect it with GFP, which he can visualize. So this is from a live animal. This is a single muscle fiber. And you can see these big blotches here are the GLUT4 protein. So this is in the resting state. He then injects the animal with insulin. The animal is obviously anesthetized during this whole thing. And then 30 minutes later, he images again. And what he sees is that these transporters, these big blotches, now become more diffuse, and we see more of the transporters on the cell surface. We also see more of a striated pattern here, and that's because muscles have these invaginations called transverse tubules, and so the transporters are there. So they go from, so the transporters, just as I showed you in the cartoon, actually move from inside the cell to the cell surface, and then they're able to transport the glucose into the cell. This is the same thing with contraction. Again, now you can see these big blotches, and you can see more of a striated pattern and more transporters on the cell surface with muscle contraction. So exercise and insulin both cause this GLUT4 translocation process in the skeletal muscle. And so one of the questions that we and many other research groups wanted to address is, are there different mechanisms for this process? So we can do this by, one of the ways we can get at this is to actually measure glucose transport. Now this isn't translocation, this is actually measuring the glucose going up into the muscle cell. So here's the muscle, it's incubated in the test tube, and then we can label it with radioactive labels, and we can measure the amount of glucose that goes into the cell. So in the basal or resting state, this is a very low, but after a maximal contraction stimulus, this increases about 3 times. And this is just what we would expect based on all the things that have been done with exercise in humans. A maximal insulin stimulation will also increase rates of glucose transport into muscle. And when you combine a maximal contraction and a maximal insulin stimulation, you get additive effects. And what this really suggests is that there's got to be 2 mechanisms, because if you take the max and you put it together, you get an even further increase. So clearly we know that there must be different mechanisms that lead to this translocation, and in fact there may be different pools of these transporter proteins inside the cell. So believe it or not, everything I just said took about 5 years, my entire PhD dissertation, to figure out, that I can sum up now in usually 1 or 2 slides, which is a little disheartening, but that's really science. So now I came actually to Boston. I was at Vermont doing my PhD where we studied this in muscle. And so now since I've been in Boston, which has been a number of years, what we've tried to understand is how does exercise cause these transporters to move to the cell surface, and how does insulin cause this also? Now, we haven't been focused on this, and a whole other group of investigators have studied this, but we've really focused on this. But what I can tell you is a little bit about how insulin works. This is overly simplified, but basically here's the insulin molecules that increase after you eat a meal. And insulin binds to its receptor, and it results in phosphorylation of the internal, or the transmembrane, but also internalized portion of the insulin receptor. This phosphorylation then leads to this complex signaling mechanism, which I've actually simplified here, but these are various proteins involved. This one is the insulin receptor substrate 1 protein, or IRS protein, leading to activation of this molecule called PI3 kinase. There's some other steps involved here, but it also leads to activation of this protein called AKT. And we know that these signals are very important for the insulin effect for GLUT4 translocation. There's been lots of genetic and pharmacological studies to show that. So when we first started this work, these molecules were starting to be elucidated. So I thought, well, both exercise and insulin cause GLUT4 translocation. Maybe some of these molecules are also part of the exercise signaling pathway. So that was about another 6 years of work to show that that wasn't true. Any way that we looked at that, none of these molecules really are important in the exercise effects. So, for example, the type of experiment that we could do is we could take a transgenic animal that had the insulin receptors knocked out, put those animals on our treadmill, and measure exercise-stimulated glucose uptake. It's completely normal. Same thing for any of these proteins that we looked at. None of these proteins, when you got rid of the activity of any of these proteins, exercise-stimulated GLUT4 translocation was completely normal. Glucose transport was normal. So then that really left us was, well, what is this mechanism? And what is the signal regulating this process with exercise or muscle contraction? And that, yep.
I was just wondering if you had looked at the cytoskeleton inside in terms of the exercise, whether that had a big contributing factor in exercise.
Yeah, so the when we first started the work and we were doing all these fractionation, the cytoskeleton was our enemy because it prevented us from getting pure fractions. We think that this is occurring, not independent, but it's not really a major factor in this translocation process. Except for some of the signaling may be common, but although now some of the other types of cytoskeletal proteins, not the ones that are contracting the muscle, but some of the non-contracting cytoskeleton proteins may be important and may form tracks to move these transporters to the cell surface. And so there's a lot of research actually going on in that now. It's really hard to do in muscle because of all these other proteins that are there, but a lot of the cell biologists that work with simplified cell systems are studying the cytoskeletal involvement in the movement of these transporters. So exercise, what is the exercise signal? What's the key signal? And this, through a long story, led us to this protein called AMPK. So what is AMPK? So AMPK stands for 5' AMP activated protein kinase. It is a heterotrimeric protein, so there's 3 subunits. The alpha subunits, alpha 1 and alpha 2, contain the catalytic activity of the kinase. The beta and gamma subunits are very interesting, and they seem to be regulatory in their function. So what is AMPK? Well, it's a member of a metabolite sensing protein kinase family, and the activity is increased, it's activated under conditions of low energy state. So exercise activates AMPK activity. And it's now been proposed to regulate numerous metabolic and transcriptional processes in multiple cell types. And this is really an understatement, because when I started working on this protein almost 10 years ago, there was about 2 papers published per year on this AMPK protein. And now I would say there's about 2 or 3 papers published a day on this protein. So it's really, you know, it's definitely one of the hottest molecules in science. It's now thought to be involved in cancer and potentially cancer prevention and all sorts of metabolic functions. So it really seems to be a key molecule in many, many different cell types and in many, many different cellular functions. But we're really interested in its role in metabolism. So I'm not going to go into a lot of detail. There's lots and lots of literature on this protein AMPK, but I just wanted to mention that, again, this is kind of how we think the protein works. This is the beta subunit is thought to be a scaffold. AMP activates AMPK, so any condition where there's low energy state and AMP increases, it can bind to this gamma subunit, which then makes this alpha subunit, which has the catalytic activity, a better substrate for upstream kinases that can phosphorylate this protein. And LKB1, which is actually a protein that was discovered to be important in cancer, and certain types of cancer, actually is one of the regulators of this protein. Okay, so what's the evidence? Why did we think that AMPK could be important in the exercise effect on glucose transport? And really what the research really came from studying this compound called ACAR. So what is ACAR? I think I have that on the next slide. Okay, so ACAR is actually a reagent that's been around for years, that's been used for various different things such as heart perfusion, etc. But what was discovered was that ACAR could be taken up into cells and it could be phosphorylated to form ZMP. And ZMP is a structural analog of AMP. So anything in the cell that's being activated by AMP will also be activated by ZMP. And so one of the things that ACAR does is it very potently can activate AMPK. ACAR can activate AMPK, this, again, is a tool that's widely used then. And I'll talk more about ACAR at the very end of the talk. So what we wanted to do was to say, okay, if AMPK is important in this mechanism, if we use this very potent activator of AMPK, will it increase glucose transport? So again, we used our system where we incubated muscles in the test tube. Here's the rate of transport in the resting or basal state. And sure enough, when we treated the muscles with this AMPK activator, we got a big increase in glucose transport. If we compared this to insulin, it was a little bit more, but not statistically different. And when we added ACAR, maximal ACAR, and maximal insulin together, we saw an additive effect. And this looked just like what we saw when we did the contraction experiments, the additivity with insulin. So then we did the experiment with muscle contraction. So here we have, again, the maximal ACAR effect. Here was the maximal contraction effect. But now when we added ACAR and contraction together, we didn't see an additive effect. And this really suggests then that ACAR and exercise could work through the same mechanism, AMPK, leading to the regulation of glucose transport in muscle. And that this mechanism is separate from the insulin mechanism. We also showed, we were measuring glucose transport, but we also showed GLUT4 translocation. Okay, so let me just summarize this part before I go on to the next part. So what we have found so far with skeletal muscle glucose transport is that exercise and insulin increase glucose transport in skeletal muscle, resulting in glucose clearance from the blood. Exercise and insulin increase glucose transport through the translocation of GLUT4 transporters to the cell surface and transverse tubules in skeletal muscle. There are distinct proximal signaling mechanisms for exercise and insulin-stimulated GLUT4 translocation. And finally, ACAR increases AMPK activity and can increase glucose transport in skeletal muscle. Okay, so let me talk now about humans and what happens in humans. Okay, so you know, this is great. This is all animal work. It's very interesting to those of us in the area, but does it really have physiological relevance for people who exercise and people with type 2 diabetes? So if we go back to our question then, based on what we know, we can kind of rephrase the question a little bit, because now we think there are different mechanisms. So does exercise bypass defects in insulin action and skeletal muscle by utilizing alternative signaling mechanisms leading to increased GLUT4 translocation? And we can actually get at this by doing studies in humans and taking skeletal muscle biopsies. Now, this is the needle that's used, and I have to say that I actually don't do this myself. I have collaborators that do that. Mainly they're in Scandinavia, because the Scandinavians don't seem to mind to have big hunks of muscles taken out of their legs. But we have lots of studies in the US going on too. I just don't do it myself personally. So basically, you get lidocaine like you get Novocaine at the dentist, and then the person will take a small scalpel and make a small cut in the leg, and then this is the needle that goes in, and it's all done under sterile conditions, and you actually use suction to help pull the muscle out, and then you get a little piece of muscle that you can see right here. Now, if this was the Scandinavians doing this, these are Americans doing this one, the muscle piece would be much bigger, because it's really quite amazing the types of things that they can do. But anyway, so we can take the muscle. Actually, I've had a lot of postdocs from Denmark and from Sweden, and one of my postdocs told me that in his laboratory, once you had 50 of these done on you, you were actually given the golden biopsy needle, and they'd take one of these and they'd bronze it and they'd give it to him as a reward. So he had one of those. So he has actually had about 50 of these done. So it's not painful, but it leaves a little tiny scar, but it's usually it really doesn't cause any problems. Anyway, so you can take these muscles and we can ask our specific question. Are there defects in insulin and exercise stimulated GLUT4 translocation in patients with type 2 diabetes? So the first question then is this translocation process. Can we measure this, and is our hypothesis correct? So here's the data with insulin. Okay, so these on the left are the control subjects. These are normal matched controls. They don't have diabetes, whereas these are the patients with diabetes. And you can see that what we're looking at is the amount of GLUT4 on the cell surface. So with insulin, you get this nice increase in the amount of transporters on the cell surface. This allows the glucose to go into the cell. And this is what you see with a control patient. But with a type 2 patient, type 2 diabetic patient, you see this is really blunted, completely blunted. Now, on the other hand, if you do exercise in these patients, you can see that it's absolutely the same as what you see with controls. So in type 2 diabetes, you get normal GLUT4 translocation. So here we have a situation then where the insulin effect is impaired in people with type 2 diabetes. You don't get GLUT4 translocation, but the exercise effect is normal. And I'm not going to take the time to show you all the data, but in a whole series of experiments that we've done on these muscle biopsies from patients, you can see defects all along the insulin signaling pathway. So all of this effect occurs, and then you get this inhibition of translocation. So we wanted to ask the question, well, if AMPK is really important, then we should be able to see normal activation of AMPK in these patients with diabetes. So we did that. And again, with one of these biopsy studies where a biopsy was taken before the exercise, and actually partway through the exercise, we stopped them, take a biopsy, and then after. And you can see this is the control subjects, exercise at a moderate intensity. AMPK activity goes up, and it stays up. And so what happens with the people with type 2 diabetes? Well, the answer is it's completely normal. Normal activation of this AMPK protein. So both in controls and type 2 diabetics, you get activation of AMPK, of the AMPK protein, the activity of this protein that we think is important in the regulation of glucose transport. And if we look at the amount of protein or the expression of protein in the muscle, it's also normal in these people with type 2 diabetes. So exercise apparently can bypass all these defects that occur in people with type 2 diabetes and can activate this AMPK protein and regulate glucose transport. So this is great. It's pretty interesting. You know, we've defined this mechanism, but could we actually utilize this to help make a drug. So could we make a drug to activate AMPK and mimic these effects of exercise on glucose transport in muscle? So this is certainly something we thought about, and believe me, once we made these discoveries, I think I went to every pharmaceutical company in the United States because they all wanted to jump on this idea that you could activate this and you could make a great drug to mimic the effects of exercise. But what we realized was, in thinking about this, is that there was this drug called metformin, and we hypothesized that maybe metformin could be working through this system. So what is metformin? So metformin is actually the single most prescribed oral anti-diabetic drug in the US. It's now used in combination with other drugs. It's also called glucophage. Its cellular mechanism of action was really unclear. We really didn't, it's been around for a long time, but no one really knew how it worked. But it's known to suppress hepatic glucose production. So in people with type 2 diabetes, one of the major problems is the liver is constantly putting out glucose. And metformin is thought to work partially by acting on the liver to suppress this production of glucose. But it's also known to increase muscle glucose uptake, and this is thought to be done by inhibiting complex 1 of the respiratory chain. So what we realized was that this compound ACAR and metformin seemed to have some very similar metabolic effects. So both could inhibit hepatic glucose production. So we'd seen that ACAR could work like metformin. They could both stimulate muscle glucose uptake, and they both could increase lactic acid production. So we hypothesized that maybe metformin was working through AMPK, both in the muscle and the liver. And I'm not going to show you the liver data, but I will show you the muscle data. So one of the first things that we did was we took this drug and said, okay, if this drug is working through AMPK, it should be increasing activity. And sure enough, if you treated a muscle from an animal for a period of 3 hours, you got an increase. How about in humans, though? In patients that take metformin, do they get an increase in AMPK? And here are the data for that. This, again, is doing a muscle biopsy experiment. So these were type 2 diabetic patients. They were washed out for 2 weeks, and then they took metformin for 4 weeks, and then out to 10 weeks. And sure enough, when people take metformin, they get an increase in AMPK activity in their muscles. And so we think that this is part of the mechanism by which metformin works to increase, to lower blood sugar levels. So clearly, we've identified a protein that can activate glucose transport and cause GLUT4 translocation into muscle. It's a mechanism that's independent of insulin, and therefore is really, there are many, many pharmaceutical companies trying to make new drugs to stimulate AMPK in muscle and in liver to have these beneficial effects. Okay, so exercise and insulin increase GLUT4 translocation in human skeletal muscle. Insulin signaling and GLUT4 translocation are impaired in patients with type 2 diabetes. Exercise-stimulated AMPK activation and GLUT4 translocation are normal in patients with type 2 diabetes. Activation of AMPK can increase glucose transport in skeletal muscle, making AMPK a drug target for treatment of type 2 diabetes. And metformin, the most widely used diabetes drugs, works through stimulation of AMPK. So these are a lot of studies, a lot of work over a period of time, and as I said, there's a lot more work going on, especially by the pharmaceutical industry, to try to develop this. But I don't want to leave you with the idea that this is so simple. In fact, what we have realized using a number of genetic approaches is that AMPK and the exercise mechanism is probably only a very small part of this signal. And in fact, probably because of conservation evolutionally, there are actually multiple signals that are probably part of this. So while we know that if you activate AMPK, you can get this stimulation, what we've also found is that if you inhibit this, some of these other molecules can take over. Now with insulin signaling, once you inhibit it, just doesn't work. But with exercise, if you inhibit one of these components, the other can take over, which also makes a lot of sense because that's why exercise is able to I think this is one of the reasons exercise has so many profound effects in the muscle. Okay, so the final The thing I'll talk about for the last couple of minutes is the exercise pill. So what is the exercise pill? So some of you may have seen this. This was about a little over a year ago now, where it was headlines throughout the world. The exercise pill has arrived. And this was actually one of my favorite quotes that came from one of the articles. It's the exercise pill, the pill that helps you burn off calories just by sitting on the couch. It's America's dream. It is America's dream. There's no question. Okay, but this was not like some far-fetched, news organization. This was the front front page of the New York Times, Couch Mouse to Mr. Mighty by Pills Alone. And the Boston Globe, our own paper right here, pills may boost endurance without exercise. It was not on the front page, but they had a big article on it. So does anybody know what the exercise pill is, actually? Well, the exercise pill is ACAR, what I have been talking about before. And so this investigator named Ron Evans actually published a paper in Cell, which is probably the highest impact journal in science in terms of basic science publications. And so it got a huge amount of press. It was also at the time of the Olympics. That probably helped. And so, just a huge onset of press, and I was interviewed by this group and that group what I thought about all this, and I'll tell you that in a minute. And so in response to this, the New England Journal of Medicine asked me to write it wasn't really a commentary, but it was taking this basic science information and making it so physicians could understand really what it meant. So the title of this, actually my title was The Exercise Pill, Modern Medical Miracle or Too Good to Be True, but they took out the modern medical miracle part. They just said too good to be true. So I guess they were a little biased. So is ACAR, can we use ACAR to treat diabetes? Is it really a great exercise pill? So what I showed you before is we know, sure, ACAR can do some good things. It can increase glucose uptake into skeletal muscle. And although this paper was published in '08, actually work from my lab and many other labs had shown this 10 years earlier. So acute effects of ACAR can increase glucose uptake into muscle. And there's been about 200 papers showing that it can lower blood glucose levels, another really good thing in terms of treating type 2 diabetes. And if you take it chronically, we showed in animal studies that you could increase exercise endurance. This was work from our laboratory, and then this was the other paper that got all the attention. But the reason we think that this happens, which actually wasn't tested in this paper, is that when you have so much glucose going into the muscle, you actually increase muscle glycogen. And I think most people know that that's what people, when they exercise, they run a marathon, they want lots of glycogen. So when you take any animal and you put a lot of glycogen, a rodent animal, and then you put them on the treadmill, they'll run forever. And that's because ACAR increases glucose uptake and you get lots of glycogen. So it has these effects to increase glycogen in the muscle. Also a good thing, especially for people that want to exercise. So here this was shown by many groups, again, quite a while ago. It also, what ACAR does is it increases the number of these transporter proteins on the cell surface, also a good thing. It also increases mitochondria and mitochondrial enzymes. So looks like ACAR is doing a really good thing. So why is it too good to be true? Well, there are many reasons it's too good to be true. First off, ACAR has a short half-life after intravenous infusion and poor bioavailability after oral ingestion. So you take it and it's gone, basically. But more importantly, ACAR treatment causes lactic acidosis in humans. That's not a good thing at all. And we know from studies in other tissues, remember I said AMPK is doing all sorts of things in other tissues, we know that AMPK activation in the hypothalamus of the brain can actually stimulate appetite. You do not want to stimulate appetite in a person with type 2 diabetes who's obese. So that's a problem. And what I think, although some cardiologists would disagree with me, I'm not a cardiologist, is that activating mutations of AMPK, so mutations in the heart that lead to really high levels of AMPK activity cause heart conduction abnormalities in humans. It's called Wolff-Parkinson-White syndrome or WPW syndrome and can lead to instant death. So not really a good thing to have happen. And I think that, so anyway, there's all sorts of reasons, and there are actually more reasons why AMPK activation using ACAR is not a good thing. I think that because there are multiple isoforms, there are probably ways to manipulate compounds to specifically activate AMPK and not have these side effects, and that's what the pharmaceutical industry is trying to do now. Not just use ACAR, but use other small molecules, specific molecules to activate AMPK, but only in certain tissues, not obviously in the hypothalamus and not in the heart. Okay, but I think the most important reason why ACAR is not a great exercise pill is shown here. There are many, many benefits of regular physical exercise. There's really strong epidemiological data, there's basic science data that exercise can do so many different things. So exercise in the brain can decrease depression and anxiety. There's now very strong data for a decrease in Alzheimer's, stroke. It can decrease appetite short term, increase in appetite, but long term, it actually can decrease appetite, improve cardiac function, improve lipid profile, lower blood pressure, increase bone density, increase muscle strength, increase flexibility. And there's good data now to suggest that exercise can decrease rates of certain types of cancer, increases pulmonary function, improves sleep, increase blood volume, pancreatic function, liver function, reduce fat stores, and prevention of diabetes. And this is what I talked about here today. So while the idea of an exercise pill, I think, is a good one, it has many, many limitations. And I think it's going to be a long time before we can come up with a pill that has all these benefits of regular exercise. So, you know, you can't sit on your couch forever. You really got to get out and you got to exercise. You can't just take ACAR and any of these drugs. So I think with that, I'll end. I just wanted to mention that this is where I work, over on the other side. This little spot right here, this is the whole Longwood medical area. And I had a nice picture, but it didn't transfer when I tried to download it, but of my lab. All these people are not in my lab right now, but these are, you can see it's a very international crew, Korea, Japan, Denmark, China, an American thrown in once in a while, and Denmark and Japan and China and et cetera, Thailand and another Danish and Canadian, etc. So all these people have contributed one way or another to all this research. We also have a number of collaborations with a group in Japan, a company right here in Massachusetts that helps us a lot in developing reagents. And then these are the ones that take the biopsies in the human subjects. And then we have funding from the NIH and some other, the American Physiological Society and the American Diabetes Association. So with that, I'll stop, and I'm happy to take any questions you might have.
You're talking about skeletal muscle with exercise increasing AMP and then glucose uptake. What about other cells? What happens when you exercise? Are there also AMPK stimulations to cause uptake of glucose in the rest of the cells?
Right. So exercise does increase AMPK activity in other cell types. So it does activate in the liver, but that doesn't function to increase glucose uptake. It activates in fat, and we think that, and although this is a little bit controversial, we think that the increase in AMPK activity in the fat actually promotes the movement of fatty acids outside of the fat, which is the fuel that the muscle needs during exercise. So during exercise, glucose doesn't go into fat. It really only goes into muscle for the most part. The other tissues don't dispose it. The liver puts out glucose, the fat puts out fat, the muscle is what takes up the glucose. So the hypothalamus, right, I talked about how AMPK activation stimulates activity, but exercise doesn't increase AMPK in the hypothalamus. And then we haven't really looked in every tissue in the body. But so I think in some places it's activated, in some places it's not, and it has a different function in every tissue.
Do you mean aerobic exercise in particular?
Yeah, so most of the research, and that's, I should have pointed that out initially, most of this type of research and the type of protocols that we use are aerobic exercise. There has been much less research in sort of signaling and et cetera on resistance type of exercise. These signaling, we've actually studied all sorts of different signaling proteins. AMPK is one of the major ones, but there's some evidence that AMPK is important in resistance exercise, in regulating buildup of muscles, hypertrophy of muscles. So, and the other thing that I think is very important is that to point out, and I need to always remember to mention this, is that there's now overwhelming data that not only is aerobic exercise important, but resistance exercise is very, very important for people, especially as they get older, for strength and mobility, and also has very positive effects on overall glucose homeostasis. So your blood sugar levels are really affected by your ability to have muscle, because the more muscle you have, the more place for the glucose to go. So resistance exercise, less well studied, but certainly we now know in terms of overall health is just as important as aerobic exercise. So, yeah.
Do we know what it is that causes the breakdown of the insulin pathways in the first place? I mean, we say that, you know, it's because we eat all this refined carbs.
Yeah.
Why are refined carbs different from, you know, like natural, whatever that is, carbs?
Yeah. So, so that's a really hotly debated area right now. There's a lot of, there's a fair amount of data to suggest, there's sort of one camp that says that the reason this insulin signaling system has broken down is because what happens is when you have all this excess carbohydrate, it actually makes fat in the muscle, and that disturbs the signaling system. There's another train of thought which is sort of linked, is that the mitochondria within muscle don't function as well in people with diabetes and have insulin signaling defects. There's yet other people that say it's a metabolic stress called ER stress that results. Some others say it's incomplete oxidation. So there's a lot of different hypotheses right now on how come this signaling mechanism is not working. And in terms of the types of sugars and, you know, why it causes more fat, it's all under sort of investigation. But it's, the bottom line is the signaling is disrupted. And I'll let those other guys figure out why. I'm going to focus on the good thing. You had a question? Did you?
I just want to, resistance exercise, you mean like weightlifting?
Yes, weightlifting, sorry. Yes, weightlifting. Yeah, very, very important. Yeah.
I have a question about, could you elaborate a little bit more about how ACAR and metformin increase lactic acid formation? So if someone is a couch potato and they would take this, if they could take it, would that make them feel like all their muscles were cramping up because of all this lactic acid?
Well, that's the thought, that if humans, because, you know, when this all came out over a year ago, people thought, oh, you know, it's going to be the latest drug doping. And I said, well, that's not going to happen because there's going to be huge high levels of lactate and you can't perform. So it was never an issue. It didn't happen. But so why is there more lactate being produced? Well, probably because when you have all this glucose going into the muscle, and you don't really, if it's really ramped up so much that you really don't have a need for it to go through the TCA cycle, you get lactate coming out. It's sort of leaking out, and it's probably to do with the liver not being able to metabolize it. So it hasn't really been extensively studied, but that's kind of the hypothesis, that you just have so much glycolysis going on that you're shunting out lactate. Yeah.
I have a question, but first I wondered where that slide came from, whether it's proprietary, that benefits of exercise.
Oh, yeah.
That's a fabulous, that makes a great poster.
Yeah, so I made that slide, but I got the image from this guy who actually, the person in that image was actually obese, and a very well-known obesity person. So if I should really talk to him, because a lot of people always want that slide. So, and I show it. I mean, he gave it to me, but whether it's okay for me to give it out, I think I should.
My question is, high blood sugar in and of itself is a bad thing. Probably I should know this. I understand the insulin, you know, if it doesn't go into cells, that's an issue. But high blood sugar itself, what's the problem?
Well, so the problem is really that it leads to complications. And so things get glycosylated and they don't function as well. So, you know, you have a lot of peripheral, you know, like blindness is a very big problem in diabetes. Now it can be treated, and so you get these molecules that become glycosylated and they don't work as well. Your liver, and then the excess sugar has effects on the liver. So the liver, so anyway, the cardiovascular and the vascular system just starts to fail because this increased glycosylation can lead to arterial sclerosis. 50 or 60%, I can't remember exactly the number, of all people with type 2 diabetes die of heart disease. So it leads to these complications. A lot of them are due to the vascular complications, and it results really from the increase in glucose. Now there's all sorts of thoughts on what the mechanism is. This increased glucose also is thought to activate this protein called PKC, protein kinase C, which is thought to have some of these detrimental effects. So it's really multi-component. You have in the vasculature, you also have effects on the liver, so your liver gets fatty and your liver doesn't work as well. So you have renal problems, again, due to the problems of the vasculature. So it's a lot of different things that are happening this increased sugar level is really causing problems. And in some cases, you get high insulin levels, and that can also cause serious problems too.
Mine's really short. Is this age independent? I mean, you probably don't have grandparents wheeling in there to have their muscles biopsy, but...
You mean age independent in terms of...
in other words, this effect.
Oh, this effect of the exercise? Well, we haven't really done studies on older people in terms of really looking at the muscle, but in whole body, you know, you can just monitor glucose in people, glucose levels in people that exercise, and absolutely in older individuals, exercise lowers blood sugar levels. There's no question about it. And resistance exercise also has these very beneficial effects. So it doesn't go away when if you turn 70 or you turn 80 years old, your muscles still need the blood sugar, so the blood sugar levels go down. So, yeah.
Just two quick questions. What percentage, like if a normal healthy person's working out, what percentage of the glucose entering the skeletal muscle goes via like the AMPK versus the insulin site? Like what percentage? And the second part would be, if it's a big enough percentage of the AMPK, could that be used to somehow treat type 1, like augment type 1 treatment?
Yeah, so the first part of the question is, so when people exercise, if you were to go out and exercise, or unless you're someone with type 1 diabetes, but if you don't have type 1 diabetes, what actually happens is insulin levels drop dramatically. And that makes sense, right? Because if your muscles are taking up glucose and you have insulin there, you're sort of having this compounding effect, and people can have very low blood sugar. You would have low blood sugar. So really, when you're exercising, the insulin signaling mechanism isn't working. In fact, we have some data to suggest that during exercise, that whole signaling mechanism is downregulated. It doesn't even work if there is insulin there. So there are different mechanisms. When people exercise, insulin levels drop, and so it's really the AMPK mechanism or the exercise mechanism that's working. Now, in people with type 1 diabetes, would this be a good treatment to take, an AMPK stimulator? I don't think so, because in people with type 1 diabetes, we think, although we don't know for sure, that the blood glucose is going into the muscles normally. So the insulin, when people with type 1 diabetes having the insulin pump or take insulin injections, the insulin system works in those people. So it's not of any great benefit to further stimulate glucose uptake into the muscle, because that's not really where the major problem is. It's more control, you know, getting the balance between the insulin and the
You can modulate it.
Yeah, I think it would probably be a little bit of a difficult way to do it. I mean, people with type 1 diabetes that exercise are always having to manipulate their insulin, the amount of insulin that they take. You know, you hear about people that exercise in the evening or late afternoon, and then their glucose levels drop. So I think it would probably be a challenging way to modulate it. Now, that doesn't mean people that have type 1 diabetes shouldn't exercise. They absolutely should exercise, because there's so many benefits of exercise, as you saw on that big, huge slide, and those things are all of benefit to people with type 1 diabetes. So.
What level of exercise are we talking about? Is it sitting at your desk all day, getting up every 20 minutes and going up a flight of stairs? Or is it?
Yeah, so the current recommendations for exercise that actually came out last year by the government, that put a big, huge panel together, had this big lead up. And then it was the day that the stock market dropped 400 points, so it didn't really get any press, which was really unfortunate. But 150 minutes per week of moderate intensity exercise is the recommendations. You can actually go on somewhere.gov and get the information. So I always say that I think the more exercise you do, the better. A lot of people feel you really should try to exercise in one way or another, 45 to 60 minutes a day at a moderate type intensity. But I think the other thing that's really important, you're saying getting up and walking up a flight of stairs. I think that just sitting all day, if you do your hour of exercise and then you just sit for the rest of the day and don't move, that's not good either. You just have to try to move as much as you can. What I try to do when I'm working, because I sit at a desk now a lot and I don't move very much, is I just try to get up and walk around or walk down a couple flights of stairs just to keep moving. The more you can move, the better. And the more you're activating these systems.
So like how long would that GLUT4 be at the membrane surface?
Yeah, so what happens is that actually reverses fairly quickly, but what happens is that when you do exercise and you do, say, a moderate intensity exercise, your muscle glycogen that we talked a little bit about before actually goes down. And then what happens is after the exercise, once the sort of the acute effects of exercise have worn out, the muscle is actually more sensitive to the actions of insulin. So, and that can last for up to 24 hours after exercise. So if you, and I had a slide in here, I don't know what happened, I guess I must have taken it out by mistake, but to showing this very important effects of exercise. So if you exercise and then an hour later you eat a meal, for the amount of insulin that you get, that insulin works better, and that is another mechanism that helps keep blood sugars lower. Yeah.
I wondered if you thought about the type 2 diabetic patients only and the epidemic in this country. If you did a cost benefit, assuming, and I don't mean to pass judgment, but that you would get a large population of these patients that aren't going to exercise, would taking the risk of taking the pill, quote unquote, whatever form it took, outweigh their long-term effects of the type 2 diabetes?
Well,
assuming that you probably have a big portion that aren't going to exercise no matter what.
Right. Well, that's why we have diabetes drugs. I mean, a lot of diabetes, if people really, I mean, I don't have hard facts, but if people really exercise and really, did an hour or an hour and a half of exercise today, probably the amount of people who are and could lose some weight along with that, the amount of diabetes that we'd have and the treatments that we would need would drop quite dramatically. But you're right, I mean, the big part of the problem is getting people to exercise. And that's a whole nother sort of story and problem. So, but that's why we have metformin, that's why we have all these drugs. There are definitely agents that can help with type 2 diabetes. I think it's very important to develop those types of drugs. But I think that what we don't want to do is we don't want to say, okay, don't worry about it. You can just sit on the couch because you're going to be able to take a drug. That's what we don't want, because there are so many other health benefits, and exercise probably works the best. It's probably the best drug. It's certainly the best preventative drug. So,
have any studies been done in people that are hypo and or hyperglycemic at the molecular level, or is it just all?
Yeah. So really, people with, you're talking about with like type 1 diabetes.
Seeing differences anatomically at the molecular level.
Yeah.
What about people who are hypoglycemic?
Hypoglycemic. So you mean people that are, you're not talking about people with type 1 diabetes, but people that have hypoglycemia. We haven't, we have not really studied people with hypoglycemia and the effects of exercise. There aren't that many people with hypoglycemia. I mean, there can be bouts that are probably due primarily to effects on the liver of hypoglycemia. There are certainly patients with type 1 diabetes that get hypoglycemic. Very little of this work has been done in type 1 diabetes because the funding agencies aren't that interested in exercise and these types of effects in people with type 1 diabetes. So,
yeah, selfish question. For all of us in the room, probably, if we want to exercise and maximize the amount of, I keep forgetting the initials, but
activation of the signaling of the glucose transport. Yeah.
What percentage of max heart rate is the optimal to exercise at? Because then obviously that's what we would aim for. It's much easier to do that than to look at oxygen uptake, which you can do in your lab, but we can't.
Well, I think that probably working out at at about 70% of VO2 max is or 70% of maximal heart rate is really probably optimal. If you're an 18-year-old kid that's trying to train for some competitive support, they can work at a higher level. But for most people, I think exercising at about 70% of maximal heart rate is good. You can, maybe not initially, but you can exercise for a pretty long period of time at that level. So I think for maximal effects, that's a reasonable amount of work, that 70% of VO2 max.
Is there a correlation though, as you increase heart rate, you increase, is it linear?
Yeah, so some of these, there's probably many different proteins that are regulating this effect, and there really haven't been systematic studies to show, okay, at this percent you get activation of this. I mean, the general thought is that the sort of the moderate intensity exercise, somewhere between 60 and 80%, you're going to have your maximal effects. Once you get up to really high intensity exercise like sprinting and that type of thing, you're actually not promoting glucose uptake into the muscle that much because you're just utilizing glycogen that's already in the muscle. So you can't you can't work at 100% for a very long period of time. Although there's a lot of people out there really pushing the idea of doing moderate intensity and then very high intensity and then moderate intensity and high intensity as a way to, not only have the fuel utilization during the exercise, but also breaking down the glycogen, which can increase insulin sensitivity. So, I think there's some validity to doing that type of workout too. Yeah.
Do you know, or have you heard of studies with the epigenomic effects? And it seems like it's increasing the type 2 diabetes, and are we passing it on to our children?
Yeah, so that's a really hot area of diabetes research right now. There's lots of people that are starting to really investigate. There seems to be some really good evidence that there are epigenetic effects, but I'm not, I haven't really kept up with that literature very well. There isn't that much literature, but there's really some thoughts that things like, there's some studies going on at Joslin right now where, again, using animal models, feeding them high-fat feeding during pregnancy, and then looking at their offspring. And it's really quite amazing because even though this can be 8 or 10 or 12 weeks, which is a long time in a mouse. They're 12 weeks after birth. There's having these effects of the mother having eaten a high-fat diet. So we're actually just starting some studies to do the same thing with exercise. If we can get the pregnant animals to exercise, we're going to see if we can reverse some of those effects. Yeah.
If you do both, take metformin and exercise, have you ever noticed there's a canceling effect? Because my wife has type 2, and she does both,
Yeah.
and her numbers are all over the place.
Yeah. So have they always been all over the place, or is it?
Yes.
Yeah.
So I'm wondering if you ever noticed in your studies, maybe there's some kind of,
Right.
you know, maybe there's an overload there or something?
So that's a question that we're really trying to address right now, and I get asked that question a lot now, because we're thinking that these are working through the same mechanism, and so the hypothesis was that if you're taking metformin and you're exercising, are you going to have even better effects? So that's the hypothesis that we're testing. We're actually working, we don't do a lot of human studies, but we're collaborating with a group out at UMass Amherst, who they're exercise physiologists, and we don't have the answer to the question right now. It doesn't look like it's additive, but we need to do more. I wouldn't think it would necessarily make things irregular. That may just be, there can be so many factors that can play into it. And certainly the subjects that they've studied so far, we haven't heard of anything like that. But there needs to be more, and he just got some research funding from the government to address this question, because it's a very important question to try to understand. All these people are, taking these 2 treatments now, and that's what's recommended. Is it having, more beneficial effects? So we don't have the answer yet, but that's what we're trying to do right now. My dad actually is a type 2 diabetic. He exercises like crazy. And he takes metformin also, but he's doing it. He's amazing. He's really doing extremely well. He has normal levels of A1C, which is, you're probably familiar with that. It's an indicator of regulation of diabetes. So for some people, it's clearly a good combination. Yeah.
The resistance to insulin, is that sort of a genetic malformation of the protein at the cell surface?
Yeah, so that kind of gets back to one of the questions earlier. What's causing this insulin resistance? And we know that the signals are not working. The proteins themselves, those signaling proteins, look to be normal. In a few very rare cases, you'll have mutations in the proteins, but for the most part, it's really thought that that's only a small percentage of what's going on, a very small percentage. And it does seem to be sort of not indirect, but other events happening in the cell, increased lipid in the cell that then is affecting the signaling. There's phosphorylation events that occur because of the lipids or because of the altered mitochondria that then disrupts the function of the signaling, the ability of the kinases to signal the other protein. So the proteins themselves seem to be fine. They really don't seem to be decreased in terms of the number of those proteins, but it's somehow the phosphorylation events get disturbed by these other mechanisms that we're still trying to understand. Anything else? Nope. Okay. Great. Thanks.